Myself, ARBAZ Khan. I'm from Mumbai. I studied in final year B Pharmacy at Srinagar Institute of Pharmaceutical Sciences, Lonavala. This topic is serum proteomics in tuberculosis. Serum proteomics is a powerful tool for understanding the complex host response to Mycobacterium tuberculosis infection. This presentation delves into a proteomics analysis of serum samples for active tuberculosis patient and their latent tuberculosis context. Objective of the study. These are the four types of objective. Identify differentially express proteins. Uncover unique proteins signature. Next is investigation functional pathway and explore potential biomarkers. First is identify differentially expressed proteins. By comparing proteins profile of active tuberculosis patient, latent tuberculosis contact, and healthy individual, the study aims to pinpoint proteins that are significantly altered during tuberculosis infection. Uncover unique protein signature. The research seeks to discover specific proteins pattern in the serum of latent tuberculosis contact that differentiate them from both active tuberculosis patient and healthy control, potentially revealing biomarkers for disease progression. Next is investigation functional pathway. The study of aims to analyze the functional pathway and biological processes associated with the identify a differentially expressed protein, gaining insight into the mechanism alter tuberculosis infection and the host responses. Next is explore potential biomarker. The search the research seeks to identify promising protein candidates as potential biomarker for early diagnosis, monitoring disease progression, and personalized treatment studies for tuberculosis. Next is participant selection and samples collection. The study included a diverse group of participants representing various economic background and demographic. Blood samples were collected from the active tuberculosis patient, individual with latent tuberculosis infection, and healthy controls. All participants provide information content before enrollment. These are the three types: active tuberculosis patient, latent tuberculosis constant, healthy control. First is active tuberculosis patient. Patients with active tuberculosis infection were recruited from the design tuberculosis clinics fulfill clinical criteria for active tuberculosis. Latent tuberculosis contact. Individual classified as latent tuberculosis contact were identified through contact tracing, exhibiting no active tuberculosis symptom, but potentially harboring the Mycobacterium tuberculosis bacteria. Next is healthy control. Healthy volunteer with no prior history of tuberculosis infection serve as control participant, ensuring a balanced comparison across the study group. Next is proteomics analysis methodology. Serum samples were subjected to a rigorous proteomics analysis pipeline employing advanced techniques to identify and quantify proteins. These are the four type of proteomics analysis. First is protein extraction, next is protein and peptide separation, third is mass spectrometry, and fourth is bioinformatics analysis. These four are very important for the proteomics analysis. First is protein extraction. Protein were extracted from the serum sample using established method, ensuring high purity and integrity. Next is digestion and peptide separation. Protein was enzymatically digested into the peptide, which were the separate based on their size and charge. Mass spectrometry analysis. Mass spectrometry was employed to identify and quantify the peptides, providing a comparative protein profile for each sample. Next is bioinformatics analysis. Bioinformatics tools were used to analyze the massive spectrometry data, identifying differentially expressed protein and their associated functional pathways. Next is differentially expressed protein in active tuberculosis patient. This graph shows in active tuberculosis patient is 0 to 1000 range. For 200, 400, 600, 800, and maximum 1000 range. The proteomics analysis revealed significant differences in protein expression profile between active tuberculosis patient and healthy controls. Protein function change in expression. First is interleukin 6, inflammation and increase. Inflammation changes in increase. C-reactive protein, also inflammation and also increase. Albumin and transferrin, these are the different. Nutrient transport, iron transport. But changes in also same, decrease. Unique protein signatures in latent tuberculosis context. The proteomics analysis identifies specific protein signature that differentiate latent tuberculosis contact from both active tuberculosis patient and healthy controls, hinting at early molecular changes associated with latent tuberculosis infection. These are the three types: latent tuberculosis contact, active tuberculosis contact, healthy controls. First is latent tuberculosis contact. Increased level of protein involved in immunoregulation and response to inflammation suggestion a heightened immunoresponse deposit the absence of clinical symptom. Active tuberculosis patient elevated level of associated with inflammation, tissue damage and dysregulation in indicative of an active infection. Last is healthy control. Protein levels within a normal range reflecting a healthy immune system and no sign of inflammation. Next is function pathway activated during infection. The differentially expressed protein identified in the study were associated with the several key biological and cellular processes, providing insight into the complex host pathogen interaction during tuberculosis infection. These are the different pathway of activated during infection, tuberculosis infection. These are the different types of pathway: immunoresponse, inflammation, tissue remodeling, metabolic process. First is immunoresponse. The study highlighted the activated of pathway involved in immunorecognition, cytokinin production, and cell signaling indicated robust immunoresponse of Mycobacterium tuberculosis. Next is inflammation. Elevated level of protein associated with inflammation, such as chemokines and cytokinin, reflect the inflammation cascade triggered by tuberculosis infection. Tissue remodeling. The study identified protein involved in tissue remodeling and repair, suggesting an attempt by the host to repair the damage caused by the bacteria. Metabolic process. Alteration in the protein related to the metabolism, particularly thus involved lipid metabolism, suggested that Mycobacterium tuberculosis infection significantly impact host energy metabolism. These pathways are inflammation for the help of tuberculosis. Protein biomarkers for tuberculosis progression. The identify differentially expressed protein hold on protein as biomarker for tuberculosis progression, earlier detection and personalized treatment study. These are the three types of potential biomarkers. First is early detection, next is disease monitoring, and third is personalized treatment. These three are the important for the potential biomarker. First is early detection. Early detection is the sum of the identified protein could serve as early diagnostic marker, enabling timely invention and improving treatment outcomes. Next is disease monitoring. These biomarkers outcome to be used monitor disease progression, access treatment, access treatment efficacy, and identify individuals at risk of developing active tuberculosis. Personalized treatment. The study suggested these biomarkers could inform personalized treatment decision, tailoring therapy based on individual patient characteristics. Next is limitation and future research direction. This study, while providing valuable insight, acknowledgement limitation in any research study. These are the four types of limitation: sample size, geographical variability, further research, further validation, functional studies. First is sample size. The study involved a relative small sample size which could impact the journalability of the finding. Geographical variability. The study was conducted in a specific geographical region, and finding may not be directly applicable to other population. Third is further validation. Further research with large sample size and across diverse population is needed to validate the identified biomarkers and their clinical utility. Function studies. Additional function studies are necessary to evaluate the prices role this patient of tuberculosis pathogenesis and host immune response. Next is conclusion and clinical implication. Conclusion and clinical implication. The study provides valuable insight into the serum proteomics landscape of tuberculosis infection, revealing distinct protein signatures, activated pathways that are critical for understanding the complex host response. Early detection and diagnosis, personalized treatment studies, further research, further direct, future research, future direction, host pathogen interaction, unique signature. The finding of this study provide novel insight into the serum proteome of active tuberculosis patient and their content. These are the important for the tuberculosis of these serum samples.